Hello and welcome everyone. In this particular section of video, I am explaining about the end replication problem, and this is also known as the termination of DNA replication in eukaryotes. Since the eukaryotes, they have a linear strand of DNA, so at the end, in case of the telomere, in case of the replication in the telomeric end, there will be some problem. All right, why is this? Because in case of lagging strand, there is a primer at the end, and after beyond this there is no more DNA, right? If in case of in case of the middle portion of the chromosome, what would happen is there are other stretches of DNA further, so the enzyme can fill the gap. But what happens is at the end there is no further DNA, right? So what happens is after the removal of primer, the enzyme, the polymerase is not able to fill this gap. All right, so there will be a gap. After the replication, after the removal of primer, there will be a gap, whereas this side there are more stretches of the DNA beyond this, so this stretch can be filled, right? But this part will not be able to filled. This part will not be filled up. It will be remaining like this at the end after the removal of the primer, because it is against the directionality of the enzyme. So there will be the formation of 3' overhang. The lagging strand, this is the lagging strand, and in the lagging strand there will be the 3' overhang, right? Whereas the leading strand is same and it is blunt. It is same, right? But the lagging strand is having a overhang part. So what can be done? What can be done is, now it has been studied that the telomere, it has a sequence of TTGGGG. Alright, it has a TTGG sequence of 1000 repeats. It has these sequence of 1000 repeats, and then this sequence is repeated 1000 times, and every time the telomere is getting shortened, this sequence is getting lesser and lesser. Alright, now what is the advantage of this particular sequence? First thing is telomere is protecting. This particular sequence is a sequence of telomere, and it is more than several thousand repeats in number in every chromosome, and this stretch is protecting the rest part of the chromosome from the nucleus degradation. First thing. Second thing is this stretch, alright, this stretch has a major role in the aging. That is the main thing. This stretch of the telomere has a major role in aging. That is why we see the difference in the skin of the old age people and us. In us, the telomere has not been shortened so much. By every division it is getting shorter and shorter, but that shortness is negligible because our age, we are not so old. But in the old people, due to the repeated number of the cell division, what happens is this telomere has become so short that it is not affording the further division. If it will, now in the old age people, if the telomere will divide more, then what will happen? The rest part will be exposed. If the rest part will be exposed, then the DNA will be degraded. So to avoid this degradation, what happens is, in the old age people, the cell division is stopped. That is why their skin is, even their skin, we can see the difference between their skin. Our skin is new because every time the cell is dividing, but in the old people the cell in their skin is no further dividing, because if it will divide further, then the telomere will be lost. All the telomere might be lost, and the DNA will be, all the rest part of the chromosome might be very susceptible, right? That is why. But But what is there is one enzyme known as telomerase. There is one enzyme known as telomerase which can avoid this overhanging, the formation of 3' overhang every time. All right, what happens is like this is the leading this is the lagging strand, for example. Now this strand is making this small strand. Now in the next cycle these will further make this much. In the next cycle this will further make this much, and then it will reduce and reduce, right? So this is a natural process and the normal cell can't do anything about this. That is why we all die one day. Our skin, our cell will stop dividing and we all will die one day. It is a natural process and it is non-stoppable. But in some cells such as cancerous cell, stem cell and germline cell, what happens is there is one enzyme known as telomerase. The telomerase enzyme can avoid this happening. How they avoid this happening is they have the sequence, they have the complementary sequence to the overhanging sequence. Alright, our telomere, it is having the thousands of repeats of TTGGGG. Alright, this repeat is more than 1000 times. For example, this. Alright, and the telomerase enzyme is having complementary sequence to this repeat. Alright, these telomerase enzyme, they are made up of RNA and protein. Now what happens is these enzyme will make a complement AACCCAAC. This is the sequence of the telomerase enzyme. Alright, this is the sequence of the telomerase enzyme and it will form a complement. Alright, and the protein part of the enzyme will be forming further nucleotide. The remaining nucleotide portion will be again be synthesized by the protein part of the enzyme. For example, this is the protein part of the enzyme. This is the protein part. Now this protein part will synthesize the further nucleotide and they will this will be compensated. Alright, and then the new strand formed by this will be same. Right, now this enzyme, since this enzyme has taken the place of the overhang and it is making the other stretch of the DNA which is in the middle, right, rest stretch which is in the middle. So now the end replication problem is compensated. This happens in the cancerous cell, germline cell or any other conserved cell. So that cell is never old. But in case of the normal living things or in case of the normal living beings, everyone get older with the passage of time because of the shortening of the telomere. Alright, why is telomere shortening? Because every time the RNA primer is removed from the lagging strand, it is not able to be synthesized in the newly formed daughter strand. So the newly formed daughter strand will be shorter every time in length. Alright, due to the repeated division of the cell and the repeated shortening of the telomere, the cell will turn older and older. Alright, and it will lose the ability to divide. But this can be compensated by this happening can be compensated with the help of the enzyme known as telomerase. These telomerase enzyme has the complementary sequence to that of the telomeric region and they are made up of rRNA and protein. The RNA part will come and form a complement and the protein part will make the further nucleotide, the gap, will compensate the gapping of nucleotide gapping, and hence the daughter strand, the daughter strand made out of a lagging strand will also have a same blunt end. Right, now this is also having a blunt end. So the daughter strand makes a same blunt end, and the end replication problem is avoided in case of cancerous or stem cell. Alright, this was only for today. If you liked my video, just hit the like button, subscribe and share my channel. Thank you.